Hello to our friends joining us via recording. Today we are wrapping up lecture lesson number 2. Before we dive into the new content that we're going to be covering in today's class, I want to do a quick moment, take some time to review one of the concepts that kind of summarizes everything that we did in class last time. And that is we're going to work together to draw and fill in a salty banana cell. Remember from what we talked about in class last time, all of the cells in your body are like salty bananas, in that if I took a banana and I sprinkled salt on it, that would represent a lot of the things like we see in our cells. So let's start on the outside of this salty banana cell. I'm sprinkling salt on it. So when I talk about having salt on the outside of this banana, that means there's 2 ions that I find outside a cell. What are those 2 ions that I find outside the cell? Do we remember which things lived outside the cell? Yeah, exactly. So on the outside of the cell, we have what's called a cation. That's a thing with a positive charge. In this case, it's sodium, my cation. And on the outside of the cell, I also have an anion called chloride. An anion called chloride. This is a banana that I sprinkled salt on. So that salt has a chemical formula of sodium chloride. That's what's on the outside. Now we go to the inside of the cells in our body. When we're inside the cells of our body, there is one ion that we have a lot of inside. Do we remember which charged thing we had a lot of inside our cells, our banana cells? Exactly. Yep. Inside our banana cells, we've got potassium, K plus, potassium. And we also talked about a couple other things that cells have a lot of inside of them. One of the things they have a lot of inside of them is glucose. Yep, like Samantha mentioned. What is glucose good for? Why do cells like glucose? What do they use it for? Exactly. Yep. Oh, check that mic for me. Getting a little bit of noise. Thank you. Yeah, so we're using our glucose as an energy source for the cell. It's their favorite food, glucose. The other thing that we have a lot of inside of our cells, though not actually inside of a banana, are proteins. So proteins, the other thing that we have a lot of inside cells. Remember that proteins do a lot of different things. So from giving the cell its shape to helping it do chemical reactions or do cool things like transport oxygen throughout the body, that's what those proteins do for us. So I wanted to take a moment to quickly review with you. Remember that we have different concentrations of different things inside and outside of our cells. Make sure we know where I have a lot of each of these things, because in lesson number 4, we're going to come back to and talk about the way that things move. So whether they go into the cell or out of the cell, that's going to come down to where do I have a lot of them later. Oops. Help me out in the chat. Can you hear me? I'm getting a message that I don't have a good connection. Okay, we can still hear me, so I'm going to ignore that. If you lose me, though, please put a note in the chat. I don't want to drop off and just keep talking for 15 minutes, because I totally would. So I'm glad we can all still hear me. All right, so we got this salty banana. We have different concentrations of things inside and outside the cell. Well, that ties into the stuff that we're going to talk about today, what Dr. Ollis likes to call the tonic words. So if we have different concentrations of stuff inside and outside the cell, we're going to compare those concentrations to each other. So when we're doing that, we have 3 words to describe the different kinds of concentrations we can have in the body. So let's start with our first concentration word here, which is hypertonic. When I talk about hypertonic, I'm talking about a solution that has more or lots of solutes. So a solution that has a bunch of salt in it compared to a solution that doesn't have a lot of salt. I would say that one with a really high concentration, I would say that is hypertonic. If I've got a solution with more dissolved things in it, that's hypertonic. If I have a solution that has a lower number or fewer solutes in it, I would call that kind of solution a hypotonic solution. So a hypotonic solution has a lower concentration of stuff in it. A hypertonic solution has a higher concentration of stuff in it. We're going to come back to this example here in a moment, but just to help us practice with this hypertonic and hypotonic thing, you can see the picture here on my screen of sweet tea and unsweetened tea. When we talk about sweet tea and unsweetened tea, this is like the drink of the South, right? So Dr. Ollis is not native to the South. I never got into the tea thing, but let's do an informal poll here in class. What do we like? We like sweet tea or unsweetened tea? What kind of drinker are you? Or are you a Coke drinker? Dr. Ollis does Coke. I don't do tea. I do coffee in the morning. Okay, a little bit of both. Yeah, raspberry tea. Oh, I like it. Yeah, no tea. I'm with you, Margarita. None, Max. Yep. So this is very interesting to me. I asked the same question to my morning class, and my morning class, a bunch of them liked unsweetened tea. So you all are my sweet tea drinkers, and my morning people apparently are the unsweetened tea drinkers. I like it. Yeah, Max is like Red Bull. Yeah, go for the big guns, right? We got to have that Red Bull. We need all the energy for sure. When I'm talking about sweet tea and unsweetened tea, the difference between these two types of tea is the amount of sugar that they have in them. So I'm comparing these things called solutes, which is what I've dissolved in my liquid. So a solution that is hypertonic has more solutes. It has more dissolved things. A solution that is hypotonic has fewer dissolved things. So in the case of sweet tea and unsweetened tea, the difference, the thing we dissolve in it, is sugar. So let's think about this for a moment. If our options are sweet tea and unsweetened tea, which of those would you say is the hypertonic solution? Based on our discussion of that word, which one would we say is hypertonic? Or to put it in easy words, which one has more sugar? Exactly. Yep. What makes sweet tea what you want to drink is the sugar that's inside of it. So sweet tea, we would say, is hypertonic to unsweetened tea. Sweet tea has more sugar in it than unsweetened tea does. But hey, just like those directional terms, it depended what order things were in, right? When I say that's when I compare sweetened tea to unsweetened tea, I'm starting with the thing that has more solutes in it, comparing it to the thing with less. I could go the other way too, though, right? So I could say unsweetened tea is hypotonic to sweet tea. So remember, just like those directional terms that we hated so much, right? Just like those directional terms, it's kind of the same deal here with hypertonic and hypotonic. The order that we're comparing things in makes a difference. In our body, we'll see here in just a moment, our body generally likes to keep things isotonic. And when we talk about isotonic, what that means is things are the same. Isotonic means the same. So hypertonic, hypotonic, and isotonic. You will hear Dr. Ollis ask you about tonic words a lot in the next couple of weeks. What I'm referring to is hypertonic, hypotonic, isotonic. Do we have more of a concentration? Do we have less of a concentration? Do we have the same concentration? That's what these words mean. Like I mentioned, our body really likes isotonic solutions. That's what our body loves the best. We want things to be the same. So when we're making things the same, there's two ways that we can do that. So let's define a little bit of terminology, hoping that this is review for us. I'm hoping we had a chance to look over this before. I'm seeing a note that there were some of us in a different meeting. Apologies about that. I'm glad you found us. I'm glad you made it back in here. Okay, so there's two ways for us to make things have the same concentration, to help us have isotonic solutions. The first way that we can make concentrations of things the same is we can move what's called solutes. So I mentioned this word on the previous slide. When we were talking about tea and we said solutes, when we were referring to the tea, we were talking about the amount of sugar. So when we talk about diffusion, if I'm trying to make my two kinds of tea the same concentration, maybe I could take the sugar from the sweet tea and give it to the unsweetened tea. That would be a process called diffusion, where my dissolved things are what moves. But the other way that I can make my concentrations the same is I can use osmosis. And when I use osmosis, I'm moving water. Now, to be technical, I'll just mention for anyone in majors biology or other courses like that, you can sometimes move other liquids besides water, but in the human body, we run on water as much as we feel like we run on coffee, right? Some of us, I'm a coffee fan. What actually moves in my body is the water when I do osmosis. So let's recap what we just mentioned. In our body, if it had its way, if it did everything all on its own, we would make things have the same concentration. Isotonic means the same. Two ways to do that. I can either move my water, which is what's dissolving things, or I can move what's dissolved inside of it. The two ways to make stuff the same. So let's look at these two processes in action. Now, I'm about to crack a really corny joke here. Let's see if you all can appreciate my corny humor. What you are looking at on your screen right now is the original YouTube. Do I get any eye rolls? The original YouTube. Okay, at least a couple of people. I know. We got a boo for groan. Yep. I know. Dr. Ollis likes to crack some bad jokes. So there's my bad joke. We're looking at the original YouTube. But hey, here's the thing. Like, you could actually watch this YouTube experiment just like you can watch videos. And it probably would be just as exciting as a cat video, no? It would not, alas. But hey, we're going to talk about how I can use these YouTubes to learn about the movement of stuff in liquids. What we're looking at in this YouTube, we have two solutions. So this is a tube that has two halves in it. In the middle of that tube, we've put a little membrane, or basically a little barrier between the two sides. So I poured solution into side one, and I poured solution into side two. And then I gave it some magic amount of time. And because it wasn't as exciting as that cat video, I came back later. And this is what it looked like in side number one and side number two. Let's do some terminology review from before. I've got two solutions here. The way that I determine who has a higher concentration or a lower concentration is by looking at the number of solutes. So solutes are the dissolved things. In this case, it's these little red dots. So not a trick question. When we're comparing the solution in side one and side two, which solution, one or two, would you say is hypertonic? Would 1 or 2 be hypertonic? Exactly. Yep, we are totally right. Side number 2 is hypertonic. Hypertonic means I have more of the dissolved things in it. Hypotonic means I have less of the dissolved things, or let me give you another way to think about this. It looks like I have more water molecules. Imagine that you are a water molecule and you're chilling out right here in the solution, and you start moving around in the water as things are wont to do. You're moving around and you're kind of taking a poll of who do I bump into? Well, if I'm a water molecule inside number 1, I'm bumping into all kinds of water molecules. Occasionally I'll bump into a solute, but I'm like, this tube is almost all water. Like, what's going on here? If I'm a water molecule that lives over here, everywhere I bounce around, I'm bouncing into these solutes. There's so many solutes over here. So here's what I'm trying to communicate. In a hypotonic solution, I've got a bunch of water. Hypotonic means there's more water than there is those dissolved things. When I'm in a hypertonic solution, there's more of those dissolved things called solutes. I've got a lot of those. So, tube number 1, this has my hypotonic solution in it. It's got more water than tube number 2. But tube number 2 has more solutes, more dissolved things in it. We've got a situation where my concentrations are not isotonic. So this YouTube is like, I got you. I can make it worth your while to watch me. We let the process of diffusion happen. And let's look at what happens with diffusion. In the process of diffusion, these little solutes, my little red dots, are like, there's a lot of us over here, too many of us over here. We're going to go next door. We're going to go into the side where there's not nearly as many of us, where we could spread out a little bit and not touch each other. So what's moving in this picture are the solutes. It's the red dots. The red dots will move over and over and over until we've gotten to a point where there's the same number of red dots in side 1 and side 2. When there's the same number of red dots in side 1 and side 2, we are isotonic. Our goal is to get to the same concentration. So one way we can do that is we can move the solutes. We can move those little red dots from one side to the other. That's possible because if you look at our little yellow line right here, this is my membrane, my fence between the two sides of the tube. Notice that it has a really big space inside of it, a space that's big enough for my solutes to pass through. Sometimes I want to put in a membrane there that it has holes that are too small for the solutes to pass through. The dissolved things can't make it in. If the dissolved things can't move, but we still have solutions that are not the same, if I can't move the dissolved things, then I'm going to move the water. I'm going to move the solvent. So now we look at which side appears to have more water. Does side 1 or side 2 look like it has more water? Which side is water more likely to bump into another water molecule instead of a solute, instead of a red dot? Ooh. We're divided, mostly landing on 2. Ironically, we actually have more water on side 1. So let's think about this. When I'm looking at my 2 tubes, like we did up above, I'm a water molecule that lives here. I'm a water molecule that lives here. When I start bouncing around here inside 2, a lot of the time I'm going to bump into all these solutes. There's so many solutes over here. If I'm a water molecule over here, I'm going to bounce around some. Oh, there's one. But look, there's a whole bunch of water. So, oh, there's a solute. Lots of water everywhere. The fact that I don't have as many solutes on this side makes it look like I have more water. Remember, this is a solution that you all told me was hypotonic. It had a lower concentration of solutes, which means it looks like it has more water. My water molecules that live over here inside 1, they bounce around and say, I am so tired of bumping into other water. I want to bump into solutes. So the water molecules travel to the other side of the tube where we have all kinds of solutes. We bumped into them in our line over here. Over time, my water molecules will keep going and going into this solution until when they bounce around in here, they hit things just as often as when they bounce around in here. When we get to the point where my solutes, my dissolved things, are as spread out on both sides as possible, now we've reached an isotonic solution. Now things are the same. So in osmosis, water will move from one side to the other. Water is going to go from where there's a lot of dissolved stuff, or excuse me, from where there's not a lot of dissolved stuff, from where there's a lot of water, into the side where there's a lot of dissolved things. Water wants to dilute the solution that's way too concentrated. I need to pause and give you a chance to process and share with me your thoughts or your questions about this. What would we like some clarity on with diffusion and osmosis? Okay, so Sharmila says she's a little confused on the diffusion part. When we talk about diffusion, we get to let our red dots, our solutes, get to move. So when diffusion happens, the things that are at a high concentration, which in this case are these little red dots, they get to go over to the other side. And they're going to go over until there's just as many of them on this side as there is on this side. When things get equal, when we're spaced out the equal amount, then we're going to stop moving. So if my dissolved thing can move, that's diffusion. Hayden asked a good question. Yep. When we're talking about diffusion or osmosis, we are always going, like Caitlin said, from high to low. So when we talk about diffusion, I'm going from where I have a high concentration of dissolved things to where I have a low concentration of dissolved things. When I'm talking about osmosis, I'm going from where I have a high concentration of water, so lots of water, to where I don't have as much water. So depending on what's moving, that's going to give you an idea of which directions things are going. Things are always moving from where there's a lot of me to where there's a little of me. Sometimes that means we're moving the red dots. Sometimes that means we're moving the water. Whatever I have a bunch of to what I don't have a lot of. So great clarifying question. Yeah, so Hayden asked another excellent question as well. Hayden asked, so how do we know if we're going to be doing diffusion or osmosis? So that is an excellent question, and we're actually going to talk about that a bunch in lesson number 4. So in the body, what helps us to decide whether we're going to do diffusion or osmosis is actually the plasma membrane. So the plasma membrane will let some things come through it. If you are a solute that plays nice with the plasma membrane, like we'll talk about in lesson number 4, if you match me and you play nice, I will let you diffuse straight through. You can come in and out all on your own. If you're not a solute that plays nice with the plasma membrane is going to be a barrier. And if I have to somehow make myself balanced with you, then what I'm going to have to do is move that water instead. So in a bit here, I'm going to show you what happens to your red blood cells when we change the concentration of something that doesn't play nice with the membrane. So I can't just move sodium and chloride in and out of my cell. So what I have to move instead is my water, because your cells always want to be isotonic. They want it to be the same inside and outside. So the short answer to all of Dr. Ollis's rambling, the cell knows which of these it's going to use based on whether or not it can pass through the membrane. And I promise there's more to come. We will definitely discuss that more. Let me scroll through my chat here. Yeah, so Jaquanda's asking, clarifying that diffusion is when the solute moves, like the salt. Yep, and osmosis is when the water moves. Absolutely. So different things move in these two processes. Victor asked for an example of osmosis. We're getting ready actually to talk about red blood cells in your body and how osmosis affects their size. You are also reading an article this week called Strange But True, Drinking Too Much Water Can Kill. And that will talk to you about how if we put too much water into our bloodstream, osmosis will happen and it will be bad for our cells. Too much water will come into the cells. So we'll see an example of osmosis here in just a moment. Yeah, and Caitlin offered the idea of we're going from where there's more of something to where there's not enough to equalize it. I love that. That's perfect. Yeah, so Hayden asked, how is it possible that these things don't require energy? Let me give you an example that would be applicable to, it's not a science example, but it's a real life example. Suppose we all come into our lecture classroom next week and I tell you all, okay, we are all going to sit in the first 3 rows of our classroom. Like, forget about COVID, forget about you being comfortable at all. We're sitting in the first 3 rows of the class. So we all get in there and we get squeezed up together. There's a very high concentration of us in the first 3 rows. You better believe that less than a second after I say, okay, we can spread out to the whole classroom, you better believe we're all spreading out as soon as possible. It's just nature. It's just chemistry. When we have things that are squeezed together, they don't like it. They always want to spread out as much as they can. So the reason this doesn't take energy is because this wants to happen on its own. We're going from where there's a lot of something, it's not happy, there's too many of me, to where there's not as much of something, down at a lower concentration. Yeah, so Lisa's mentioning if we mess with our chemistry by doing something like drinking soy sauce, or the flip side, drinking too much water, absolutely, you can have a medical crisis. So go light on your soy sauce. Absolutely. Yeah, so Hayden mentioned it comes down to a word that we're going to talk about a bunch in class next week and the week after. Our membrane is semi-permeable. It's only going to let some things do diffusion. Everything else, if we have to balance it, we're going to do osmosis, and that can be challenging for our red blood cells. They can expand too much or contract too much. We're hoping that we stay more at balance and don't really mess up our red blood cells. Glad you liked the example, Shanti. I've thought about that one quite a bit. Yeah, so when we use the word semi-permeable, it just means that some things can get through, some things can't get through. So the things that can get through, we'll use diffusion for. Anything that can't get through, if I have to make it equal, I'm going to have to use osmosis to try to make it equal. Awesome. So I want to take us back to our tea because we were all really excited about the prospect of sweet tea. Another way to help us make this talk of diffusion and osmosis make more sense, I think, will be if we use this example. So let's use a concrete example together of sweet tea and unsweetened tea. So I'm going to help you out in filling in some of these parts of your notes outline. We'll do this here together. We've already talked about this, but we'll do a quick review. When I'm talking about sweet tea and I'm talking about unsweetened tea, was it sweet tea or unsweetened tea that we said was hypertonic? Hypertonic. Who has a higher concentration, basically? Yeah, absolutely. My sweet tea is what has a higher concentration of sugar, hypertonic, which means that my unsweetened tea would be the opposite word, right? My unsweetened tea would be hypotonic. It has less sugar, or if we're thinking about it in terms of water, it has more water. So let's do this together. I'm going to pull an Olive Garden, if you will. Whenever you go to Olive Garden, they have that grater, right, where they give you Parmesan and they're like, just tell me when to stop. It's like, oh, keep going, all the cheese. So here we go. Y'all are going to help me since most of us drink sweet tea. Y'all are going to help me know how much sugar to put in. So tell me when I've drawn enough sugar in our YouTube. Here comes the sugar in our sweet tea. And again, you tell me when we have enough sugar. We love our sugar. Let's be real, right? Sugar. Does this look about right if we just filled it with sugar? That'll do. That's good. Yeah. Perfect. Okay. For I think I only had one or Texas sweet. That's right. That is Texas sweet. Yes. I can't remember if I had anyone who drinks unsweetened tea. For any of my unsweetened tea friends, do you put any sugar in your tea or no? No sugar. Whoo, man. Okay, Roxana does a little bit. Lisa and Hayden are a no-go. Well, here's what we'll do for the sake of helping us understand. We'll say that there's one little naturally occurring sugar in there. We'll put one little sugar. Yeah, so just not the sweet tea level. Yeah, I get that. Sweet tea is real sweet. So here's our two solutions. And we kind of already talked about this before, right? How we have a ton of sugar in sweet tea. We can see it all. And we have very little or even no sugar in our unsweetened tea. So when I'm talking about the solute, that's the dissolved thing. That's the sugar. And you guys accurately told me that sweet tea has more sugar in it. It has more of the solute. That's why I call it hypertonic. And you accurately told me that because I have less sugar in my unsweetened tea, that's hypotonic. I've got two solutions side by side in this YouTube that are not isotonic. They're not the same. So there are two ways that I can move things to make them isotonic, to make it equal. I can either move my sugar from one side to the other, or I can move my water from one side to the other. If I'm going to use the process of diffusion to make these two types of tea isotonic, am I moving the sugar or the water? If I'm doing diffusion, which one would move in diffusion? Exactly. When I do diffusion, I'm moving the dissolved thing. So in diffusion, I'm moving my sugar from where there's a lot of sugar to where there's a little bit of sugar. So in the case of sweet tea next to unsweetened tea, this sugar is going to say, whoa, there is way too much of us over here, not nearly enough of us next door. The sugar is going to go out of the sweet tea and it's going to go into the unsweetened tea. The sugar is going to go from high concentration, Texas sweet high, right? Concentration, to lower concentration. So if that's what happens in diffusion, then my other thing over here is osmosis. It's the movement of water. When I talk about the movement of water, remember that water is going to go from where it looks like there's a lot of water to where it looks like there's less water. When I'm looking at my two sides of the tube over here, which side looks like it has more water in it? Which side is going to have water molecules that bounce around and hit each other instead of hitting solutes? Yeah, exactly. My tube on the right, right? Over here, I've got a bunch of space all throughout this tube, a bunch of space where there's no sugar or very, very little sugar. That means that my water is going to want to go out of my unsweet tea and it's going to go into my sweetened tea. Because I have so much water on the unsweet tea side, not nearly as much on the sweetened side, I'll send that water over to help spread out those sugar molecules to try to make it equal isotonic on either side. And I think we can probably agree that if we were to actually make isotonic tea, pretty sure that nobody's going to drink that, right? We either need all the sugar or we need none of the sugar. There's not really a happy medium. We just get yucky tea. So when you're thinking about the two ways that we can make things have the same concentration, either we can move the dissolved stuff or we can move the liquid. They're each going from where there's a lot of me to not as much of me. That's why it's considered passive transport. Let me pause and open the floor for questions or clarifying thoughts. Anything we'd like to ask about the tea. Isn't that a phrase, right? What's the tea? I'm not cool enough to say things like that. Yeah, so Alyssa asked a good question. Are we always going to balance? In general, yes. If I can do passive transport on something, so if it's something that can go in and out of a cell's membrane, we will always be doing this process. So we're always going to balance things that can go across the membrane. Yeah. So I'm getting ahead of myself a little bit here, but when we were talking about things like sodium and chloride and potassium, how they weren't equal inside and outside, with those things, we can't actually do diffusion and we can't actually do osmosis. That's the only way it's possible for us to have different concentrations. Because if we could do diffusion or osmosis with something, we're going to end up equal. So it's only the kinds of things that we can't do diffusion and osmosis with. Those are the only things that won't be equal. Everything else will be equal. Absolutely. Jamel says that I can pretend I'm cool enough to say that. I appreciate that, Jamel. Okay, so let's take our example from tea to actual real life in people. You may feel like your bloodstream is filled with tea, right? But we know a little better. Oh, here's a good review question for us. We're looking at red blood cells here, and red blood cells are inside a fluid. What is the name of the fluid that is outside of red blood cells, but inside blood vessels? Outside red blood cells, inside blood vessels. Exactly. Yep, we're talking about a fluid called plasma. When we talk about plasma, the fluid that I find outside of my red blood cells. In general, in your body, we want our plasma to be isotonic, meaning the concentration of the liquids that are in our plasma outside our cells and the cytosol that's inside our cells, we want the concentrations to be the same. Because if my concentrations are the same, some of my water molecules will say, well, I got to check it out inside. I got to figure out what it's like in there. And just as soon as they come in, some of the water molecules that are inside are like, oh, I got to go outside. I got to see it out there. The rate of water coming into our red blood cells and the rate of water coming out of our red blood cells is the same. So when we're talking about saline, that's the example of what's in your lab packets. If you go to the emergency room and you're dehydrated, they'll talk about giving you saline because we have a salt water solution inside our cytosol. The concentration of salt that's inside our cells is 0.9%. 0.9% sodium chloride. So if we want to make the concentration of salt inside and outside the same, we have 0.9% inside. An isotonic solution, isotonic plasma, would also be 0.9%. Now, sometimes we can get into a situation where there's so much water outside our cell that instead of any water wanting to leave the red blood cell, water only ever wants to go in and in and in. That's what we see in our picture over here. If enough water goes into your red blood cells, not only are they going to lose their nice flat appearance that we see here, so not only are they going to get puffy, they might even actually explode, like we see on this one here. When you are reading the article about drinking so much water that it can lead to death, this is the idea behind that. If there's so much water inside your bloodstream, that's going to force water into your cells. And when it forces enough water into the cells, they will expand and possibly explode. Now, help me out. What kind of solution would we call it if there's way more water on the outside? Would a solution be hypertonic or hypotonic if it has more water, if it's sending all the water inside the cell? Or about 50-50 split. Well, here, let me show you my other option and see if that helps us, because we keep going back and forth. The other option would be we put our cell in a solution where there's less water outside. The water that's inside the cell wants to leave to go dilute it. So if I have a solution out here that doesn't have as much water as we have inside, or if I have a solution that has more solutes, more dissolved things, this would be an example over here of the hypertonic. A hypertonic solution has more dissolved things, which makes that water want to leave the red blood cell. If water is rushing into my cells, it means that I have less salt outside than I do inside. This is an example of what would happen in a hypotonic solution. A hypotonic solution. So remember, let's recap a few important things. Hypotonic means that I don't have as many dissolved things. That's what the name technically means. I don't have as many dissolved things. If I don't have as many dissolved things, it looks like I have more water. So if I have more water on the outside, it wants to go inside. Hypotonic solution. Over here, I have a hypertonic solution. In a hypertonic solution, I have more dissolved things outside than I have inside. Those dissolved things, because of their chemistry, those dissolved things couldn't sneak inside in this case. But what could happen is the water in my cell can sneak outside. Because remember, we want to be equal inside and outside. So if I put it in a solution where there's a bunch of dissolved things, that cell will give away its water to try to make outside and inside the same. When we look at these cells, a cell put in a hypertonic solution will shrivel up and get tiny. A cell put in a hypotonic solution will swell up and explode. Neither of those shapes of our red blood cells, neither of those anatomies matches the regular anatomy that we see right here in an isotonic solution. Not a trick question. Here's what I want to know. For cells that are shriveled up because they were in a hypertonic solution, or for cells that are exploding because they were in a hypotonic solution, do you think they can still do their job? Their anatomy has changed. Do you think they can still do their physiology? Yeah, exactly. Not nearly as well, or especially if we're exploding, right? Definitely not at all. So here is an example of our idea from last week, right? Anatomy and physiology are related to each other. If my anatomy changes because my blood is too salty, that's what's going on over here, my blood cells are going to shrivel and they might be more likely to get clumped up and stuck in places in my blood vessels. Or if my blood vessels are huge or my blood cells are huge because I have too much water, those cells might start exploding and then I don't have enough cells to carry oxygen. So isotonic, the concentrations are the same inside and outside. That's good for your cells. By the way, I got a good question based on this slide. The question was asking me about what numbers, like what salt percentages we would say would be hypertonic or hypotonic. And you can have any answer for things that are hypotonic or hypertonic as long as it's either above the normal concentration, that's hypertonic, right? Or it's below the normal concentration, that's hypotonic. So when you're looking at your notes outlines, there are a couple of different hypotonic solutions that are listed there for you. One of them, 0.45%. Another one of them, 0% pure water. Both of those kinds of solutions have a lower number than the normal number. But really, hypotonic could be as simple as 0.8, right? Anything that's below normal, that's hypotonic. Your notes outline talks about 3.5%. 3.5% sodium chloride. That's an example of a hypertonic solution. But again, anything that's above 0.9 is hypertonic. So 1%, 1.5%, the specific numbers don't matter. It's just always either above normal or below normal that we would say is hypertonic or hypotonic. I need to pause and I need you to help me know how I can help you. What would we like to clarify? Lisa, when you're talking the plus or minus sign, I'm not quite sure what you mean. When would we use the plus or the minus? Well, she's typing, yeah, if we're talking about charges, yes. If we're talking about a charged molecule, yes. When we're talking about sodium chloride together, we don't have the plus and the minus, but if sodium's by itself or if chloride's by itself, we would need to have the plus or the minus. Yeah, and somebody had mentioned in the chat, what's the concentration inside? Multiple people are correct in saying that the concentration inside is 0.9%. That is accurate. Yes, 0.9%. Let's see. So we have an interesting application question. So Jamel asked if we had too much water in our blood. So if we had made ourselves hypotonic, could we drink soy sauce to save ourselves? Interesting question that, I mean, it maybe could work. I think it would be smarter probably to go to a hospital. But in a pinch, I suppose it might help. Yes. Let's see. Yeah, so we're just following up on the percentage, 0.9%, because that is the percentage that we have inside our cells. When we have that outside our cells, that is isotonic. That's correct. So that's where that number 0.9 came from. I'll try to put it in a little cloud here. 0.9% is the concentration inside your cells. So when it's outside your cells, it is considered isotonic. Great questions. Yeah, I need to check out Chubby Emu. I know you've mentioned that a couple of times, Lisa. I haven't looked into that one yet. I have kind of two more ideas that I want to hit on before I split us up into groups. Here's the first idea that I want to hit on with you here. Somewhere in your notes, possibly on like the last page of your notes, you have this statement here. And this is an underline, highlight, star statement. This is a really important idea. The idea is water follows salt. Water follows salt. This is the simplest description of how osmosis works. Water is looking around in the tube that it's inside of, and it's saying, is there a lot of salt over here, or is there not a lot of salt over here? If water sees that there's not a lot of salt in the place where it's at, water is going to go find where there's more salt. So when we were looking at those red blood cells and we were looking at the direction that the water was moving, if it was leaving our cell, it's because there was a higher concentration of salt outside the cell. If it was going into the cell, it was because there was a higher concentration of salt in the cell. So when you're looking at osmosis, wherever we have more of those dissolved things, water is like, I want to get in there. I want to do this. So when you're thinking about osmosis, short and sweet, it's water following salt, moving toward the dissolved thing. So in osmosis, we're moving from where there's a lot of water, a hypotonic solution has lots of water, and it's moving into a hypertonic solution. A hypertonic solution is one that has a lot of salt or a lot of sugar. Yeah, and so I know that some friends have already helped us out in the chat, but the clarifying question is we will always determine hypertonic and hypotonic based on the two solutions. If we are above the normal or below the normal, that's absolutely correct. Yep. And on quizzes and exams and even on today's group work, I'm going to give you some random numbers, and it's just straight up comparing numbers. Which number's higher, which number's lower, that's hypertonic or hypotonic. So it is as simple as that, even though those are weird words, right? It is as simple as that.
Professor, does this go along with the plasma at the end of our worksheet, or is this water follows salt different than the plasma?
That's correct. Yep. So this is the same. You have it in the context of plasma in your cell, inside the body. So into and out of red blood cells, it works just the same. I put up the picture of the YouTube here, but it's the same thing with plasma. So it applies the same whether we're in a YouTube or whether we're talking about red blood cells. Yeah. Great question.
Okay, thank you.
Mhm. So just to break down, let me see, to break down that statement from the previous page, I want to remind us here, when we talked about water being the thing that's moving, remember that's osmosis. So in osmosis, water moves, and water is going from where there's lots of water, lots of H2O, into where there's little water. Or the other way we can compare a hypotonic solution to a hypertonic solution is where we have a little of the salt, a little of the dissolved stuff, to where we have lots of the dissolved stuff. Two ways to think about osmosis. Either water is going to go from where there's, let me get my pointer here. Water goes from where there's lots of water to where there's not as much water. Or it's going from where there's not very much salt to where there's a lot of salt. Different ways to think about that statement that we had on the last page there. So in osmosis, however it's easiest for you to predict the way that water moves, these sets of statements are true every time. Find a way that works for you and study it that way. So I see a question in the chat. Will we be moving water instead of solutes most of the time? It depends on the particular solute that we're talking about, but for the most part, if we're talking about things doing their own natural thing where we don't have to use any energy for that, yes, it's usually going to be water moving. We'll talk a little bit next time about how we use the process of diffusion to help us with breathing. So the way that we get oxygen into and out of our cells is actually using diffusion. So that's an example of diffusion, but we don't do too much diffusion in our cells because our plasma membrane is kind of like a bouncer at a bar, and it's not going to let very many things inside. Any other last minute questions before I turn off our recording and split us into groups? Don't forget, as we're typing, don't forget your guided lesson to help you fill in any of the holes in your notes outline. We did not cover everything. So you will need to use that guided lesson to help you round that out, and make sure to read that article about drinking too much water.
Professor, I have a question.
Mhm.
When it talks about what direction does it move, so we're going back up to the YouTubes. It asks what
Mhm.
direction does osmosis move,
Uh,
and then asks a whole bunch of other questions on like, in what way does water move or something like that. What is that?
So that would be something that the guided lesson will help you out with, but I'll give you a general idea. When we're talking about the direction that things move, they're pretty much always going to move from high concentration to low concentration. So they're moving from where there's a lot of themselves to where there's not as much of themselves. And the guided lesson will go into that a little bit more in depth. But with what we're talking about, which is passive transport, we're always going from high concentration to low concentration.
Okay, so it's not like a left or right type thing, it's a high and low.
Yeah, big picture, yes. When we're talking about the YouTubes, we can say like left and right. But in your body, there's not really a left and a right, right? So in your body, we're going from high concentration to low concentration.
Okay, thank you.
Samantha asked about CPR. I don't know, you'd have to kind of explain that to me a little bit more, how that could be an example of that. I don't understand the analogy, but perhaps there is one that could be made there. Let's see. How much water would you need to drink to kill yourself? You're going to read about that in the article, so I won't spoil the article for you, like Hayden said. Yeah. All right, well, I am going to turn off the recording. I'm going to get those groups set up.